Our next and last speaker will be Shanting Wu. She is from the Wen Lab at the University of Hong Kong, and we will go back to zebrafishes. So I look forward to hear her talk. So the floor is yours.
Okay, thank you. So everybody can hear me clearly?
Yeah.
Okay, let me change this to laser point. So good morning, good afternoon, good evening, and everybody. So I'm Shanting Wu from the Hong Kong University of Science and Technology, and I work on the microglia using zebrafish. So today I'm glad to share with you my research work, which is about the heterogeneity of microglia in the zebrafish adult brain. So I think the importance of the microglia do not need further introduction, but for those of you who are not familiar with microglia, there's one thing that I want to emphasize is that they were extremely versatile phagocytes in the central nervous system. So you can see that in addition to function as an immune cells to remove the cell debris or the invading pathogens, they are also very critical for the development and the function of the neural circuit. Here shows that the microglia actively remove the undesired synapse, a process called the synaptic pruning. So here it's not surprising to see that the dysregulation of microglia is actively involved in the pathology of the neuron degenerative disease and the psychiatry disease. So one problem that have been bothered the researchers in this field for a long time is that how is this functional versatility of microglia being regulated? So one answer to this question is that whether there's any possible heterogeneity inside the microglia population, which means that basically different subtype of the microglia fulfill different function. So actually, 100 years ago, when a microglia was first identified by Ortega, he already observed the different morphology of the microglia shown here. And at that time, he was trying to link this different morphology to the functional phenotypes. But I have to say that even after 100 years, we still do not have a very clear answer to the heterogeneity of microglia. So nowadays, thanks to the advanced single cell analysis, we can probe the microglia transcriptome states from the single cell level. So here, several researchers have already done the single cell RNA analysis in the mouse microglia and the human microglia. But interestingly, they found that in the mouse adult microglia, it was a rather homogeneous population without very obvious subpopulation. While for the human single cell analysis and mass spectrometry analysis, they found that there was greater diversity. More specifically, here you can see that they found differentially expressed genes in the gray matter and white matter microglia, and also they also found a difference in terms of the regional in terms of the regional differences. So because most of the microglia studies are focused on the mouse, and we believe that the research involving a diverse species can provide important insight in the functional diversification of microglia during evolution. So that's why we work microglia in the zebrafish. So here we have identified two microglia populations in the zebrafish that have distinct morphology and distribution by accident finding. So initially we were looking at this mutant that was called the CSF1A mutant, which is short for colony stimulating factor 1 receptor A, a very important growth factor for the macrophages. So previously I have already shown that this signaling pathway is very important for the development and the colonization of the early microglia precursors. But later, when I chased this phenotype to the larval stages and adult stages, I noticed that the remaining microglia in the CSF1A mutant showed a very interesting morphology and distribution pattern. You can see here that they were all very ramified cells with a lot of protrusions, and they also enrich in the center of the midbrain slice, which is actually enriched for the white matter. So when we go back to check that in a wild type fish, we found that these similar kinds of ramified microglia can also be identified in similar region, but at the same time, there was another kind of microglia which have more amoeboid morphology, indicating that they have bigger cell body but less protrusion. And this kind of amoeboid microglia can be found throughout the brain. So we were fascinated by these two kinds of microglia, and we went on to search for any specific markers that can only mark one population of this microglia. And luckily we got this marker which called the CCL34-beta1, and which is a chemokine. And we found that this gene can specifically mark the amoeboid microglia. So here you can see that we generated a transgenic line with the CCL34-beta1 driving the expression of the eGFP, while at the same time we use the MPEG-1 that's read to mark all the microglia in the brain. And here you can see that the GFP was expressed in this widely distributed amoeboid-shaped microglia, but it was negative in this ramified microglia that was enriched in the middle of the midbrain, so indicating that the CCL34-beta1 was specifically expressed by the amoeboid microglia. So through the FACS experiment, we can find that the CCL34-beta1 positive microglia is the predominantly microglia population, accounting for 80% of the total microglia, while the ramified microglia accounts for only 20%. So now that we already have these two transgenic lines that can distinguish and separate these two populations, we are able to perform the RNA sequencing. So that's what we did. And firstly, we performed the principal component analysis with these two microglia populations, together with some other immune cell type, such as the Langerhans cell in the skin, neutrophil, and T cells, to see their relative relationship. And we found that these two microglia populations, they separated as two distinct clusters, but they have a further distance compared to other immune cells, indicating that these are two microglia populations in the adult brain. So we further checked their key transcription factors because we want to find out the differentiated regulating pathway, and we found that both microglia population express many common transcription factors for the macrophages development, such as the SPI1 and the IFA, while at the same time they also express their own transcription factor in very high level. Here you can see that the CCL34-beta1 positive microglia express the transcription factor like JUNB and the phosphate, which are actually immediately early genes for neuron, or the stress response genes and the pro-inflammatory genes for the macrophages. And interestingly, for the CCL34-beta1 negative microglia, we found that they express many transcription factors that are important for the development of the dendritic cells, such as the ID2A and the BATF3, indicating they may have some function of the dendritic cells. So we further go on to check their differentially expressed genes and do the geo-turn analysis. And we found that the CCL34-beta1 positive microglia was enriched in the lipid biosynthetic process, regulation of T cell activation and a scavenger receptor, while the CCL34-beta1 negative microglia was enriched in actin binding, immune response, and cytosolic calcium ion concentration. And I think based on these geo-turns, we can suggest that the CCL34-beta1 positive microglia maybe in immune alarm stages and actively involved in the phagocytosis and the membrane transformation, while the CCL34-beta1 negative microglia, on the contrary, was more active in the cytokine and the chemokine signaling pathway. So with these transcriptome differences, we want to further check their differences in terms of the cell behavior and the dynamics. So we turn to this in vitro brain slide cell culture. And firstly, we will focus on their mobility. So here you can see that the CCL34-beta1 positive cells here shown as the red yellow cells here, they were very actively moving around the brain slices during the imaging area, while the CCL34-beta1 negative microglia were relatively immobile, staying in their original position, but at the same time they were still extending and retracting their process all the time. So the next, we check their phagocytosis capability using the E. coli bioparticles. So first, we incubated the CCL34-beta1 GFP transgenic fish with the red E. coli bioparticles. So here you can see that after 4 hours incubation, many of the GFP positive microglia have uptake the red bioparticles and become double positive. But the next experiment, when we incubate a double transgenic fish with the green E. coli particles, we will expecting to see that when this CCL34-beta1 negative microglia, if they uptake any green particles, then they will become double positive. But this turned out to not be the case. We found that we didn't observe any single ramified microglia showing here uptake any green particles, indicating that this CCL34 beta-1 negative microglia have relatively lower phagocytosis capability of the E. coli particles. So next, we further check their function in the inflammation by establishing a CNS infection model by directly injecting the E. coli into the cerebral ventricles of the zebrafish and then collect their brain for the cryosection after 6 hours. So here you can see that different from the control where the CCL34 beta-1 positive microglia were distributed evenly across the whole brain, we found that in the E. coli injected fish, they were significantly accumulated in the ventricle region where the E. coli was spreading along the brain ventricle, and also they were actively phagocytosing these E. coli, indicating they respond significantly to the E. coli infection. While at the same time, we found this CCL34 beta-1 negative microglia, they were remaining in this previous position, and their number also do not change very significantly. So we still want to further look at their transcriptome responses in terms of the information. So we performed RNA-seq also after 6 hours of the E. coli injection, and we found that actually both cell populations respond to the E. coli infection at 6 hours. It's just that the amoeboid microglia respond more significantly than the CCL34 beta-1 negative microglia. So if we take a look at their upregulated genes separately, we will find that the CCL34 beta-1 positive microglia upregulated many classical pro-inflammatory cytokines, such as the TNF-alpha and the interleukin-1 beta, while the CCL34 beta-1 negative microglia shown here upregulated a very interesting cluster of the chemokines, which is called the CXCL11 family. And they have already been reported to be involved in the recruitment of the T cell and the macrophages to the infection sites. So based on this data, we propose that the CCL34 beta-1 positive as the phagocytotic microglia, which are actually highly mobile and also have high phagocytosis capability and mainly executed the tissue clearance function. And also we named the CCL34 beta-1 negative microglia as the regulatory microglia because they were relatively contain low mobility and the phagocytosis capability, and they mainly release the immunoregulators during the inflammation. So this is the last of my talk. Finally, I want to acknowledge my co-authors and my collaborators and anyone who have borrowed our reagents, and any questions are welcome. Thank you.
Thank you very much, Shiting, for the wonderful talk. And I think it's kind of, well, here we have seen a different kind of motility. I'm not an expert in the field, but I could find some resemblance to what Sylvia showed us before. And so let's see. Okay, we have a question from the audience, from Elena. Do you have any idea when these two microglial cells types appear in development?
Okay, so I'm not sure whether Elena is familiar with the development in the zebrafish, because actually zebrafish have two distinct ways of the microglia. One is called the embryonic microglia, and the other is called the definitive microglia, which means the microglia in adult. And we found that actually the embryonic microglia only have one population, that was the CCL34 beta-1 positive phagocytotic microglia, while for the adult microglia, we found they have these two population of microglia, and CCL34 beta-1 positive microglia was always there since the embryonic stages, while the CCL34 beta-1 negative microglia will appear a little bit later. Like for zebrafish, it was around 10 to 15 days post-fertilization. So I hope that answers your question.
I'm sure that answered the question, and Elena thanks you in the chat. So if we have another question from Yunlu. Fantastic work. Do you know if CCL34B.1 positive MG exists in mice? mouse Mg are less mobile than that in fish. Is it possible that we have been looking at the other population?
Well, thank you for the question. Well, this is a very difficult question because actually we cannot find very direct counterparts for these two microglia in a mouse. And as I mentioned in the introduction, the single cell RNA sequencing of the mouse microglia indicates that the adult mouse microglia are rather homogeneous population without very obvious population. But indeed, in terms of the morphology, the microglia can be generally divided into the amoeboid shape and the ramified shape. So that's why we think that even though the mouse do not have very clear counterparts to our two populations, but our two populations still represent the two functional states of the mouse microglia. And at the same time, even though the homeostatic microglia was rather homogeneous, when there's some pathology, either the inflammation or demyelination, other kinds of pathology, they will find that the microglia will have higher heterogeneity. They will have more activated microglia that have similar amoeboid shape or even upregulated pro-inflammatory chemokines, something like that. We think that this transcriptome and morphology changes in the homeostatic microglia also indicates that they will have some functional shift, and we hope that the two populations in the zebrafish can provide the functional diversification hints in this general field of the microglia activation in the pathology. Yeah, thank you.
Thank you very much for answering this difficult question. And so I think we have to close this this great session. I would like to thank you again, Silvia, Shooting, and Daria for their wonderful talks, and all the attendees that followed us here.